90 papers this week, 6 continents, and one theme running through most of them. Let's start in Brazil with drought. Brazil has just come through a once-in-a-century yellow fever outbreak. The trigger was not rain, it was the lack of it. A new analysis in Science Advances links that outbreak to drought dynamics across the region and warns that climate change is loading the same gun again. Drought does something quietly dangerous. It concentrates water shrinks back to a few pools. Hosts crowd in, biting insects crowd in with them. Contact goes up and not one new species has to arrive for that to happen. Cuba next, a separate team modelled where dengue can spread today and where the climate will let it spread tomorrow. Their goal is a bioclimatic warning system, a forecast, not a post-mortem, and climate does not stop at the insect. It reaches into the chemistry. This is Culex quinquefasciatus. The southern house mosquito, researchers exposed it to deltamethrin and to malathion across different temperatures and humidities. The two insecticides did not behave the same way. Warmth changed the dose, which means a bioassay run in a cool laboratory may not describe the village at all. In northwestern Ethiopia, in Metema district, another group followed, where Aedes mosquitoes rest as the seasons turn. It sounds like a small question. It is not. If you do not know where they sit, you do not know where to spray. Now, the vectors that moved. Anopheles stephensi, an Asian malaria vector, and an unusual one because it breeds in tanks and cisterns and construction water. It is a city mosquito. Researchers have now characterized its biological forms in the city of Djibouti in the Horn of Africa. Knowing which form you're dealing with is a difference between a control plan that works and one that misses. Ixodes scapularis, the black-legged tick. A new report from the CDC tracks its pathogens pushing south into western North Carolina from November 2024 through August 2025. The tick is not new to America. The pathogens arriving with it are new to those counties. One of the things that tick can carry is Powassan virus. A new study in mice followed the infection into the brain and asked what it does to the blood-brain barrier on the way through neuroinvasion and the inflammation that follows it. Then China, Guangzhou City, 2025, a chikungunya outbreak, now written up in full epidemiology transmission and the vector behind it. Chikungunya is the arbovirus to watch this year. Japan published monthly and yearly trends in imported cases going back to 2011. Turkey described a traveller returning from Cuba with joint pain that would not settle. And in Spain, a clinic screening migrants who had gone home to visit friends and relatives found a hidden burden of dengue that nobody had counted. Travel medicine is now surveillance. So what about the tools? Anopheles funestus. In Western Kenya, it is pyrethroid resistant, and it is the reason a standard net underperforms. Western Kenya is where a new net was tested. Miranet Combi carries 3 actives. One kills, one blocks the enzyme she uses to survive the first one. One stops her offspring ever reaching adulthood. And a modelling group asked a sharper question about nets in general, not how many are hanging, but how many are still working. They call it efficacy adjusted use. Coverage and protection are not the same number, and the continent's maps have been quietly conflating them. Elsewhere, a solar-powered spatial repellent device went through laboratory trials, and in Western Kenya, a social science team asked the people living with one whether they liked it and whether they believed it worked. Both answers matter. A device nobody keeps switched on protects nobody. In Ixodes, a controlled trial ran an attractive lethal ovid trap, a container built to invite an egg-laying female in and not let her leave. Ixodes sits deep in the Peruvian Amazon dengue country at Aedes aegypti. The target of most of this week's chemistry, toxic sugar baits, were tested against both insecticide-resistant and insecticide-susceptible populations. The lethal effects differed, so did the sublethal ones. Resistance does not just change whether she dies, it changes what she does if she lives, which brings us to where resistance comes from. A striking paper this week put malaria vectors through generational selection with environmental pollutants, not insecticides, and watched resistance evolve anyway. The transcriptomes told the story. Agricultural and urban pollution is a selection pressure in its own right. Behind a great deal of that sits one enzyme family. A historical review looked back over 4 decades of cytochrome P450 research across Africa. Who did it, what it found, and where it has to go next. Anopheles coluzi, a malaria vector of West Africa, infected with the fungus Metarhizium pinctates. It does not simply die. The effects carry across life stages and into the next generation, biological control with a longer shadow than the dose, or Bacchia now. The bacterium that makes mosquito matings fail, so eggs never hatch. In Medellin, Colombia, researchers measured Wolbachia prevalence in Aedes aegypti across 5 communas. That is not the release, that is the part everyone forgets, checking years later whether it's stuck. Another team took a different route entirely. They raised antibodies against a chimeric peritrophin antigen, a protein of the mosquito's own gut lining, and fed them to her in blood. Her survival dropped a vaccine that protects you by killing what bites you. Plasmodium falciparum, the parasite itself. In Science this week, chemovaccination with a late liver stage antimalarial produced durable immunity. Let the parasite reach the liver, hold it there, and let the immune system learn the hard lesson properly. And it is not all mosquitoes. Sandflies, the vector of visceral leishmaniasis Kala Azar in Malda district, West Bengal. 5 years of program data show what sustaining elimination actually looks like after the headlines have gone. Black flies, Simulium. They carry the worm that causes river blindness. 2 drug papers this week went after the worm's bacterial symbiont rather than the worm high-dose rifampicin with albendazole in people and coralopyronin A in mice. Kill the symbiont and the worm stops reproducing. Triatomins. The kissing bugs of Chagas disease, a survey mapped their fauna and distribution across the Amazon to Cerrado transition zone, the surveillance gap that outbreaks tend to fall through. And Culex, the biting midges carrying Oropouche virus in the western Brazilian Amazon and Bluetongue in central Thailand, where a 3D printed suction trap with ultraviolet light and carbon dioxide is doing the catching. 90 papers, every one of them linked below. See you next week.